Human Immunity by Waugh. Infection occurs when the body is invaded by microorganisms that can cause disease and illness. There are many ways for these pathogens to enter the body, but typically it's through the respiratory tract, the digestive tract, or the skin. Infections can remain localised to the point of entry or can spread throughout the body. The body can be infected by different microorganisms such as bacteria, viruses, fungi, and parasites. Infections can be transmitted through direct contact with another person or animal, or between mother and child in utero. Indirect contact occurs when an infected person touches an object or surface and the pathogen is picked up by another person who is subsequently infected. Infection through a vector can occur when a person is bitten by an insect and a disease enters the body. Ingestion of food or water that contains harmful bacteria can cause serious illness. The first line of defence against infection is provided by the innate immune system. Physical and chemical barriers stop pathogens from entering the body, causing infection. We will look at specific examples of the first line of defence. Firstly, the skin. The skin provides one of the first lines of defence to potential infection. Formed of 3 layers, it is a waterproof and mechanical barrier that stops pathogens before they cause infections. The epidermis is the outermost layer and is made up of cells that produce keratin. It continually replaces itself over a 4-week cycle. The epidermis contains lymphocytes and Langerhans cells that create an immune response and play an important role in fighting infection. The gastrointestinal tract is one of the most important areas of the body in regards to immunity, with 70 to 80% of the body's immune cells found in the gut. Stomach acid and intestinal enzymes destroy pathogens. A single layer of semi-permeable epithelial cells allow the body to uptake nutrients but provide a barrier to disease-causing pathogens. The peristaltic movement through the digestive tract helps to remove potential infections from the body. Damage to the lining of the gut or an imbalance in gut flora can lead to pathogenic organisms entering the body. The female genital tract mucosal surfaces contain antimicrobes, and the pH of the vagina, which is normally between 3.8 and 5, prevents growth of pathogenic organisms. The urinary tract has a number of ways of protecting the body from infection. The urethra provides a physical barrier that helps stop bacteria entering the bladder. Females have shorter urethras than males, making them more susceptible to urinary tract infections. Micturition flushes out pathogens from the bladder, and epithelial cells that line the urinary tract secrete antibacterial agents. Mucosal membranes are found all over the body, including the nasal cavity, mouth, lungs, and gastrointestinal tract. They stop pathogens entering the body but allow nutrients to be absorbed. They are composed of epithelial cells that secrete a sticky mucus that provides a protective covering to the delicate cells underneath. Mucous membranes trap unwanted particles and also contain antimicrobial peptides. An injury will cause inflammation, a protective, non-specific response from the innate immune system. Inflammation will only occur in vascular tissue, and its purpose is to help seal off the injured area, stopping infection from spreading, as well as starting off the wound healing process. When an injury occurs, resident immune cells such as macrophages and mast cells release chemicals such as histamine, prostaglandins and cytokines, which cause blood vessels around the injury site to dilate. This increases circulation to the area, and this will cause the area to appear red and hot. Endothelial cells retract in the cell wall, allowing plasma and proteins known as exudate to enter the site. This is what causes the clinical sign of swelling. Kinin proteins are activated, causing the nerve cells in the area to become sensitive, which causes pain. Phagocytosis is the process by which cells such as macrophages, neutrophils destroy and engulf pathogenic bacteria and damaged cells. Both cells are formed in the bone marrow and form part of the inflammatory response to infection. Neutrophils are the first phagocytic cell to arrive, and each can destroy 5 to 20 pathogenic cells. They are short-lived and die after approximately 5 days. The macrophage arrives later and lives much longer, around 2 to 3 months. macrophage can destroy 100 pathogenic cells. When discussing the third line of defence, it is important to note the differences between a pathogen and an antigen. To recap, a pathogen is an organism that causes disease and takes on the following forms of bacteria, virus, fungi or parasites. An antigen forms part of the pathogen and is a chemical signal that tells the body it is being attacked by a disease or infection. They can take one of the following forms: proteins, carbohydrates, fats and nucleic acid. The third line of defence is known as acquired immunity. Acquired immunity is not present from birth. Acquired immunity is a learned response from exposure naturally or artificially to different antigens. The body creates a targeted immune response which remembers the antigen and can prevent further infection from the same microorganism. The body's ability to remember a specific antigen is the key in differentiating between the innate immunity and acquired immunity. Neutrophils account for 50 to 70% of leukocytes and are found in the circulating bloodstream. They are the first white blood cells to arrive at the site of infection and work by phagocytosis. Monocytes account for around 15% of circulating white blood cells and are turned into macrophages when they migrate to peripheral tissue. They kill pathogens through phagocytosis. Eosinophils account for only 1% of leukocytes in healthy humans. They play an important role in the elimination of parasites and the allergic response. They spend a short time in the circulating blood system before migrating to the thymus and the GI tract. Basophils are the least prominent of white blood cell. They have a short lifespan of only 1 to 2 days. They are agranulocytes and contain enzymes such as histamine, which is released during an allergic reaction. They are considered immune surveillance cells and can target early cancer cells. The immune system is dependent on binding molecules called immunoglobulins. They are also referred to as antibodies. They recognise and bind to specific antigens and can be soluble or membrane bound. Each immunoglobulin is made up of 4 polypeptide chains consisting of 2 heavy chains and 2 light chains. These are arranged in a Y shape with the top of the Y forming the antigen binding site. There are many different types of immunoglobulin. IgG, the only type of antibody to cross the placenta, the most prevalent, accounting for 75 to 80% of all antibodies. The smallest immunoglobulin and can be found in the tissue and bloodstream and promotes phagocytosis. IgA, the second most prevalent antibody, are vital in the role of mucosal homeostasis and is found in the secretions of the mouth, the gut, respiratory and urinary tracts. IgM, known as macroglobulins, are the largest of the antibodies. They account for 6 to 10% of immunoglobulins. IgMs are the first responders to infection and begin the attack on pathogens. IgE respond to parasitic infections and are involved in the allergic response. High levels can be found when the body overreacts to allergens, such as in hay fever and food allergies. IgD is the least well known of the immunoglobulins and is found on the surface of B cells with a very small free floating in plasma, linked to the activation of mast cells and basophils. The purpose of our immune system is to fight pathogens. Our body needs to be able to differentiate between self, which are our own cells, for example red blood cells, and what a pathogen or non-self is. This is known as self-tolerance and is crucial to balance appropriate immunological defence and normal physiological function. Pathogenic antigens are recognised and remembered by B cells, triggering antibodies to be produced when recognised. All cells in the body have a unique set of markers on the cell surface that define the cell as self. These markers are called major histocompatibility complex molecules 1 and 2. MHC1 is found on all nucleated cells and creates an immune response against endogenous antigens. MHC2 is found on antigen-presenting cells that bind and present to CD4 and T cells, creating an immune response to exogenous antigens. Depending on how different the pathogenic antigen is to our own cells, will define how strong an immunological reaction our body creates. Failure to self-tolerance is caused when lymphocytes develop receptors to self-antigens and the body fails to destroy them in the bone marrow or thymus, and they enter the secondary lymphatic system. Hypersensitivity is an immunological overreaction to stimulus that causes cell damage and inflammation and can vary in type and severity. And examples include allergic reaction to pollen, which triggers hay fever symptoms, reactions to receiving incompatible blood, and dermatological reactions to substances such as latex. Autoimmunity is the immune response to self-antigens, and autoimmune diseases are caused by this response. The exact cause of autoimmune conditions is still unknown. However, scientists believe there are contributing risk factors such as gender. Approximately 80% of people who are diagnosed with an autoimmune condition are women, diet, genetics, age, and environment. Type 1 diabetes is a chronic autoimmune condition that stops the pancreas producing insulin. Insulin is a vital hormone that controls the amount of glucose in the bloodstream. Without insulin, our body cannot break down glucose, and this leads to hyperglycemia, which can be fatal. Symptoms include excessive thirst, weight loss, fatigue, blurred vision, increased urination, and bedwetting in children. Diagnosis is achieved through testing blood sugar levels in people who present with symptoms. Treatment for type 1 diabetes is long-term insulin, either through injection or a pump. Multiple sclerosis is a chronic autoimmune disease that occurs when the immune system attacks and causes demyelination of the sheath that protects the nerves. This damage causes interruption to the nerve signals from the brain and the rest of the body. There are two main types of MS. Relapsing and remitting MS is characterised by episodes that flare up and then improve over time, with progressive MS symptoms getting worse over time with little or no improvement. Diagnosing MS is complicated and there's no definitive tests. Doctors will rule out other conditions that may cause similar symptoms. MRI imaging is used to identify plaques of demyelination, but doctors require two separate episodes of symptoms to give definitive diagnosis. Symptoms of MS include vision problems, pain, spasms, numbness, pins and needles, cognitive impairment, bladder and bowel problems, and difficulty with speech and swallowing. There is no cure for MS, and treatments involve slowing the progression of the disease and treating symptoms. Ocrevus is a 6-monthly infusion used to slow the progression of MS. It targets specific marker CD20 on the surface of infected B cells and destroys them. Tysabri is another infusion used to slow down MS. It keeps T and B cells in the circulation and blocks them from entering the central nervous system. The transfer of an organ from one person to another can be a life-saving operation. However, immunological rejection will occur in all patients except identical twins. The antigens on the cells of a transplanted organ will be treated as non-self by our immune system. To combat rejection, tissues must be matched as closely as possible using tissue typing and anti-rejection drugs such as monoclonal antibodies and other immunosuppressants. These stop the B and T cells attacking the transplanted organ cells. Types of rejection. Hyperacute, a life-threatening acute immediate rejection of the transplanted tissue or blood product. The transplanted organ must be removed or the recipient will die. Acute can occur within 1 week to 3 months post-transplant, and symptoms include pain, swelling, fever, increased blood pressure and decreased urine output. And chronic, prolonged immune attacks against the transplanted tissue over years, causing organ damage. Acquired immunity is not present from birth and can be subdivided into active and passive. Active immunity is created by us, our body's immune response to a pathogen. Active immunity can be further split into naturally acquired, exposure to a pathogen in the environment, for example a virus or bacteria. The body creates an immune response to the pathogen by T cells targeting cells and B cells remembering the specific antigen, and this creates long-term immunity. Immunity can be lifelong in cases of disease such as chickenpox and measles. Viruses such as colds and norovirus have shorter lengths of immunity. It is believed that immunity to norovirus can last between 6 months and 2 years. Artificially acquired. The body creates antibodies to a pathogen through vaccination. A dead or weaker version is injected and the immune system response is similar to that of natural immunity. Passive immunity can be subdivided into natural and active. Natural passive immunity occurs when antibodies, in particular IgG, cross the placenta in utero. Immunity is also passed between mother and child during breastfeeding. Natural passive immunity is not long-term and lasts approximately 4 to 6 months. Artificially acquired passive immunity occurs when a person receives antibodies from an immune individual. This does not stimulate the immune system to fight off the disease, but the loaned antibodies provide short-term immune response. The first recorded case of immunoglobulin transfer was in 1895 when it was used to treat diphtheria. Vaccines contain harmless forms of pathogenic antigens and stimulate the individual to create antibodies. The intention of a vaccine is to create immunological memory. When a vaccine is injected into the body, an inflammatory response from the innate immune system causes dendritic cells to take up the antigen and migrate to the lymph nodes, where they present the antigen to the helper T cells. This is the beginning of the primary specific adaptive response. The T and B cells bind to the antigen, and the T cells release cytokines which stimulate the B cells. These B cells differentiate into plasma cells and memory B cells. Antigen-specific antibodies are produced fighting off the infection, and the antibody levels peak and then contract. Memory B cells produce continuous low levels of antibodies. When a vaccinated individual encounters the pathogen, the body creates the secondary immune response. Memory B cells quickly activate and differentiate into antigen-specific effector cells that target the pathogen in a stronger and more rapid response. The principle of herd immunity is that enough people are immune to a pathogen through being vaccinated against the disease. It stops the disease spreading to those who do not have immunity. Herd immunity and vaccination programs have been used to wipe out many diseases in the United Kingdom, such as smallpox and polio. Herd immunity requires a specific threshold of how many people are immune to a particular disease to be met in order to be created. For example, according to the World Health Organization, herd immunity against measles requires about 95% of the population to be vaccinated. The remaining 5% will be protected by the fact that measles will not spread amongst those who are vaccinated. Due to a growing global population and international movement, immunity to contagious disease is very important. The 2020 COVID pandemic caused widespread death and severe illness. Scientists were tasked in developing a vaccine very quickly and rolling it out over the global population in an unprecedented short amount of time. It was hopeful that herd immunity would be achieved as a way of ending the pandemic. This, however, so far has not been the case. COVID-19 has a high reinfection rate and has changing strains of the disease. There has also been a reluctance from people to be immunised due to early side effects of some vaccines. Vaccines have been able to improve symptoms of sufferers and limit reinfection, but not stop it completely. As previously discussed, one of the most recent vaccination programmes has been to COVID-19. According to the gov.uk website, over 151 million vaccines have been given in the United Kingdom, and approximately 80% of adults have received one dose of the vaccine. Factors affecting the uptake of COVID-19 vaccines have been the lack of information on the vaccine safety, especially in groups such as pregnant women, accessibility to vaccine centres for vulnerable patients who are shielding, and concerns over side effects. The UK has one of the most robust and free immunisation programmes in the world, with childhood disease vaccination starting at 8 weeks to 14 years old. A variety of diseases such as measles, mumps and diphtheria, polio and HPV are vaccinated against. The polio vaccine was introduced in the 1950s, with the last outbreak of the disease during the late 70s, and the last recorded case in the UK in 1984. There are many factors that affect the uptake of immunisations. According to the gov.uk, barriers to attending vaccination clinics can include transport difficulties, language and communication difficulties. The National Institute for Clinical Excellence have recommended some strategies to improve access to vaccinations, including providing longer appointment times, walk-in vaccination clinics, services offering extended hours, and mobile and outreach services. One of the most damaging vaccination safety controversies was the 1998 study by Andrew Wakefield that suggested a link between the MMR vaccine and inflammatory bowel disease and autism. This caused widespread concern amongst parents and led to a significant decrease in the vaccination uptake. The study was discredited. According to the World Health Organization, Immunisation is a key component of primary healthcare and an indisputable human right. Vaccines are also critical to the prevention and control of infectious disease outbreaks. They underpin global health security and will be a vital tool in the battle against antimicrobial resistance. Vaccination has led to the eradication of some diseases, such as smallpox in 1979, a significant decrease in child mortality, cervical cancer prevention with the introduction of the HPV vaccine in 12 and 13-year-olds. Overall, vaccination prevents 3.5 to 5 million deaths per year.